The overall goal is to provide a simple assay to measure gap junctional intercellular communication through a population of mammalian cells that can serve a broad range of in vitro pharmacological and toxicological needs.
This method can help answer key questions in the toxicology field, such as whether a compound affects the development or maintenance of a tissue by dysregulating gap junctional intercellular communication. The main advantage of this technique is that it offers a simple and quick assessment of gap junction function in a large population of cells in response to toxicants and toxins. Demonstrating the procedure will be Aina Azuni, an undergraduate research assistant from my laboratory.
To begin the experiment, seed 3 × 10⁵ WB F344 rat liver epithelial cells onto 35 mm diameter culture plates containing Eagle's modified medium plus 5% fetal bovine serum. Culture the cells in an incubator until they reach 100% confluence, and continue with the experimental treatment of the cells. To continue with dose-response experiments, add 10 microliters and 20 microliters of a previously prepared 2 millimolar stock solution of phenanthrene in 100% acetonitrile, and 4 microliters, 6 microliters, and 8 microliters of a 20 millimolar stock solution of phenanthrene in 100% acetonitrile to 3 plates of cells containing 2 milliliters of growth medium for each added volume of stock solution. Next, add 4 microliters, 6 microliters, 8 microliters, 10 microliters, and 20 microliters of a 100% solution of acetonitrile.